Hello everyone, wishing you all a blessed day, energized day, vibrant day. So in our previous class we have discussed regarding the mitochondria, ribosomes, plastids, chloroplasts and its functions, various functions we have discussed. So today we will be discussing from the cytoskeleton to microbodies. So what is the cytoskeleton? We know that किसी भी सेल का फ्रेमवर्क तैयार करते हैं ये जो साइटोस्केलेटन. ओके, जैसे कि बोन फ्रेमिंग द फ्रेमवर्क, नो, इन द सेम वे द सेल्स, द स्केलेटन, द फ्रेमवर्क ऑफ द सेल्स, दैट इज द मेन फंक्शन ऑफ दिस साइटोस्केलेटन. जनरली दे आर नेटवर्क ऑफ filamentous and the proteinaceous structure. Okay, they are of proteinaceous. And जो कि जैसे कि माइक्रोफिलामेंट्स, माइक्रोट्यूब्यूल्स, एंडोप्लाज्मिक रेटिकुलम जो कि साइटोप्लाज्म में प्रेजेंट है. So we know that microfilament is of 6 nanometer while the microtubules is of 25 nanometer. So what is microfilaments? We know that a composed, these are composed. Okay, so they will be contractile. Okay, what is it? Microfilaments are contractile. They are made up of contractile protein. They are made up of actin. Okay, what is the use of these microfilaments? They are used for the muscle contraction and these are usually hollow. Microfilaments are usually hollow. And what about the microtubules? Microtubules, we know that it is made up of tubulin protein. A tubulin protein से बना हुआ है. And this is solid. ये solid होता है. When the microfilament is of hollow, we have learned just now. Yeah, this is of solid. And what does it help for? It helps for the formation of cilia and flagella. It helps in the formation of centriole. It helps in the formation of spindle fibers. ये जो है, this will be for helping for the cilia. Cilia और flagella का formation में मदद करते हैं. Centriole का formation में मदद करते हैं. Spindle fiber का formation में मदद करते हैं. These are helping for such activities. Okay, then what are the main functions of the cytoplasm? If we ask, that it is mainly mechanical support. Okay, mechanical support, motility. What is the meaning of motility? Movement, moving from one place to the another. And maintenance of the shape of the cell. Okay, so how to maintain the shape of the cells. All these are the functions of the cytoskeleton. Now moving forward. Here is the image of a cytoskeleton. So moving forwards with the cilia and flagella. Okay, cilia and flagella. What do we understand by cilia and flagella? We know that cilia means they are the outgrowths, hair-like outgrowths of the cell membrane. Okay, cell membrane का outgrowth होते हैं and are covered with the cell membrane. This will be covered with the cell membrane. We can call it as the hair-like outgrowth of the cell membrane, cilia. And it is small structures work like oars. जिस तरह पतवारे होते हैं जहाज में, इसी को बोलते हैं oars. Okay, small structures working like oars. Okay, oars means पतवारे. Okay, जिस तरह पतवारे जहाज में होते हैं उसी तरह ये एक्टिंग करते हैं. Then causing the movement of either the cell or the surrounding fluid. So causing the movement of the cell or the surrounding fluid, whatever it is, it will be causing the movement. Next, prokaryotic flagella, that is of monofibrilla. Okay, then emerging from the centriole-like structure. Where is this emerging from this cilia? From the centriole-like structure called basal bodies. From the basal bodies it is being emerged. And it is not covered with the membrane. Okay, it is not covered with the membrane. And cilia का जो core है उसी को क्या बोलते हैं? Axoneme. Okay, core or filament is called as axoneme. Then it is composed of microtubules. Okay, parallel to the law. So you can see here, composed of, we know that cilia is composed of microtubules. Then they are arranged of microtubules. 9 plus 2 is the arrangement. The 9 microtubules in the peripheral region and doublet. They are doublet, while 2 microtubules will be in the central and other as a single. Okay, 2 microtubules in the central and the other are the single. Then connection between the central tubules, that is called as bridge. Then covering of central tubules, that is termed as central sheath. Then connection between the peripheral tubules, that is termed as link. Okay, so core of the filament, that is termed as axoneme. Then it is composed of microtubules, that is parallel to what is the arrangements 9 plus 2. Then 9 microtubules in the peripheral region, it is doublet, while 2 microtubules will be in the central region and the single. Okay, connection between the central tubules, it is termed as bridge. Covering of the central tubules, that is termed as central sheath. And connection between the peripheral tubes, that is termed as link. And the connection between the peripheral and the central tubules, that is termed as radial spokes. Okay, connection between the peripheral and the central tubules, that is termed as radial spokes. And flagella, in the case of flagella, we know that it is comparatively longer and responsible for the cell movement. Cilia and flagella both are for the motility, movement from one place to another. Cilia we have seen what is it, and flagella also we have seen. Okay, so core filament is termed as axoneme. That's all. We are moving forward to centrosomes. Now let us learn about the centrosome and centrioles. Okay, centrosome and centrioles. These are perpendicular to each other. आपस में ये दोनों perpendicular होंगे. Okay, so they will be perpendicular to each other. Centrosomes, two centrioles, perpendicular to each other, and surrounding amorphous pericentriole materials. What is the meaning of amorphous? Amorphous means we can't separate it. Okay, जिसको separate नहीं कर सकते उसी को बोलते हैं amorphous. So present, they are present in the perpendicular. Okay, next is of we will centriole. Okay, they are cylindrical structure without membrane. First we will learn what is the main function of the centriole. What is the main function? It helps in the formation of spindle fibers during the cell division. It helps in the cell during the cell division, helps in the formation of spindle fibers in animal cells. The main function of the centrosome and the centriole is that formation of the spindle fibers during the cell division in the animal cells. And the next one is formation of basal body of cilia and flagella. Flagella or cilia का basal body जो है उसी का formation. Okay, that is the main function of this centriole. Okay, then it is absent in almost all the plant cells. Okay, in the plant it is present. Centrioles are present only in the animal cells. We know that. Then cylindrical structure without membrane. We know that there is no membrane. We have learned in the previous class that non-membranous one, membranous one and non-membranous one. We have learned ribosomes, basal body and the centrosome are non-membranous, isn't it? We have learned it. Then in transverse section it appears as cartwheel. Okay, चक्की की तरह appear हो जाते हैं, जैसे घोड़े की गाड़ी है जो चक्की है उसी का जैसा cartwheel. Okay, then it is composed of 9 peripheral microtubules. How many? What is it composed of? 9 peripheral microtubules. The centrioles are composed of microtubules, then of tubulin protein. Okay, 9 peripheral microtubules of fibrils, then of tubulin protein, which is composed of. Then the centriole, peripheral tubules, they are triplets. Then central proteinaceous part, it is termed as hub, and connection between the hub as well as the peripheral tubules, that is termed as radial spokes. And the arrangement of microtubules in the centriole is non+0. Okay, once again I will explain. In transverse section it will be appearing like a cartwheel. Okay, what is cartwheel? We know that चक्की की तरह घोड़े की गाड़ी जैसे वो चक्की की तरह appear हो जाते हैं. Okay, so it appears as cartwheel. Then composed of 9 peripheral microtubules of fibrils as well as the tubulin proteins. And peripheral tubules, they are triplets. Central proteinaceous part it is termed as hub, and connection between the hub as well as the peripheral tubules is termed as radial spokes. And what is the arrangement of the microtubules? That is 9+0. So moving towards the microbodies. Before that, just we will see the image of the centrioles. As I have told you, centriole, how it is formed? It is formed of microtubules. You can see here in the image. This is the microtubules. Okay, it is composed of microtubules. Now moving towards the microbodies, the last part of this chapter. So what are microbodies? We all know that they are membrane bound. Okay, they are membrane bound, and it is called as vesicles. Okay, microbodies are membrane bounded. It is called vesicles. Various enzymes is present here. Okay, various enzymes are present in the microbodies. Okay, they are found in the plant cells and also they are found in the animal cells. And they are divided into 3 types. What are they? Spherosome, peroxisomes and the glyoxysomes. Okay, spherosome we know that mainly it is found in the plants. So lipid storage का काम ये करते हैं, और digestion plant में जो lysosomes, that is also done by the spherosome. Okay, more in cotyledons and endosperms. Okay, helping in the oil seeds like castor, store of metabolism of fat. Okay, all these are the main functions of the spherosome. We can say that spherosome mainly found in the plants. ओके लिपिड स्टोरेज में इनका काम होते हैं, डाइजेशन प्लांट की डाइजेशन में लाइसोसोम्स का काम. ओके ऑल दीस आर द फंक्शंस ऑफ दिस वन. नेक्स्ट मूविंग टुवर्ड्स द peroxisomes. Peroxisomes we know that it is present in the animals as well as in the plants. Okay, so beta oxidation of the fatty acids, we know that fatty acid का beta oxidation ये जो peroxisomes करते हैं. Okay, it also has enzymes and also H2O2 metabolism in animal cells. Metabolism is taking place, and this also peroxisomes also contains enzymes. It helps in the photorespiration. And with the function chloroplast and mitochondria. In photorespiration, the peroxisomes function with which cell organelles? Chloroplast as well as the mitochondria. Then glyoxysomes. These are the largest microbody. Okay, these are the largest microbody. We know that it is found in the fatty seeds as well as in fungi. In fatty seeds and fungi only we can see these glyoxysomes. And it helps in glyoxylate cycle. So what is the main function of this one? It helps in glyoxylate cycle in which fat will be converted into carbohydrates. इसमें fat जो है carbohydrates में convert हो जाते हैं. So what is microbodies? We know that they are membrane bound and it is called as vesicle. Okay, these microbodies contain various types of enzymes. Okay, these microbodies are present both in the plant cells as well as the animal cells. What all they include? They include spherosome, they include peroxisome, they include glyoxysome. So what is spherosome, what is peroxisome, what is glyoxysome we have seen. So spherosome it is present in the plants we have learnt. Okay, peroxisome it will be having enzymes, metabolism में function करते हैं. These will be helping in the photorespiration. Okay, all these are the functions of the peroxisomes. As I have told you, beta oxidation of the fatty acids. Fatty acid का beta oxidation के लिए भी ये काम करते हैं. Then the glyoxysomes, the largest microbody we can say. In fatty seeds and the fungi only it is present and it helps in the glyoxylate cycle. So with this we completed our chapter here. So please go through it. If there is any doubt you can ask me. So please go through your NCERT text. Go through the PDF also. Prepare the notes for it. So thank you for being attentive. Thank you. May God bless you all.